Good morning, everyone. It's 2 after, so we're going to go ahead and get started. So I'm just going to give just a couple of very brief remarks, and then Dr. Liz Kovacs will come up and give the formal introduction. Dr. Billiar, Dr. Tim Billiar, is here today as part of this grand rounds and also our 3rd annual Department of Surgery research seminar, which will be at 10 a.m. in the Don Elliman conference room at the AHSB. So all of you who are not in the operating room and can make it, please come. It's really going to be a great event. Dr. Billiar is, simply put, one of the most distinguished leaders in American surgery. If you look at any metric, whether it's number of papers, presidents of society, NIH grants, mentees that he's trained, titles and leadership positions that he holds, he is at the very top and extremely distinguished. I'm going to ask Dr. Liz Kovacs to come in and fill those details in a little bit, but we are so lucky to have Dr. Billiar here today, and thank you.
So for me, it's truly an honor to have Tim Billiar as our guest speaker here for Grand Rounds and having him stick around for our research day. He was our first and only choice for the year 3 Department of Surgery Research Retreat. Dr. Billiar started his life in Iowa. He went to medical school at the University of Chicago Pritzker School of Medicine and did surgical residency at the University of Minnesota, continuing on in Pittsburgh, where he rose through the ranks to the level of scientific god, as well as distinguished professor, and as you can see here, the George Vance Foster Professor and Chair of the Department of Surgery. He has so many awards and accolades, including Distinguished Service Awards and Lifetime Achievement Awards, that it is truly remarkable where he has come from and where he has gone to and where he still is going. And we're honored to have him here as our speaker. He is the PI of a T32 in Pittsburgh, as well as an R35, and has had funding since forever, I believe. And with that, I will allow him to come to the podium. Hope he comes to the podium to give his awesome presentation. Thank you, Tim.
Well, I really can't tell you how thrilled I am to be here. It's truly a great honor for me. I'm a little embarrassed by some of the remarks that were made about me because for me, this is why we do this, an opportunity to come to another institution and share ideas and learn. I will say one thing about the titles that I've accumulated, just so you know. I didn't ask for a single one of those. They've been given to me, and some have been a blessing, some have been a curse. But it's I think an indication of a institution that wants to create opportunities for the people that they want to keep. And I've been there for a long time, 38 years. I transferred from University of Minnesota to University of Pittsburgh, not because I was getting kicked out of Minnesota, which would be the normal interpretation, but I was following my mentor, Richard Simmons, with the idea that I was going to be in Pittsburgh for 2 or 3 years, finish my residency, and 38 years later, a phenomenal wife and 2 kids. It's really worked out. Well, I'm going to talk about trauma. I don't need to tell this audience anything about trauma on the clinical side. The most recent review that talks about the importance of trauma came out this year from Peter Ree, and it really reestablishes that trauma is a huge healthcare problem. It's the unmet need pandemic, if you will, and it's increasing in incidence I don't know why poisoning deaths get thrown in with trauma, but even without that, the incidence per capita is increasing. It's third overall. And according to this review, it's the most common cause if you add up all the causes now to 67. I mean, obviously, if you're in your 60s, there are other problems that way, as I'm learning, but it is really common. And it's the leading causes of preventable deaths are bleeding and organ dysfunction. And, you know, understanding these problems are where the great opportunity is to improve survivorship. I like this paper from Karen Brohe, now nearly 10 years ago, where they just surveyed what was happening in trauma patients, about 600 trauma patients. And you can see three patterns emerging: patients with very little MODS, patients with moderate MODS that they recover from, and then these patients that are very sick and linger in the ICU. MODS peaks early in trauma, within the first 2 to 3 days. So you have an endpoint really early on that if you could modify that, we could change the outcomes. At least that's what Dr. Cohen and I believe and talk about frequently. Now, if you go to other disease processes like cancer or even processes like aging, they'll talk about the hallmarks of the disease, right? So we've decided to put together something called the hallmarks of trauma, which tries to capture at a high level all the elements that go into thinking about outcomes in trauma. So this is our figure and process. It'll end up in a paper eventually. This is the core of the figure, and it starts with the pre-injury state. Now, you all know this, of course, but there are fixed factors such as age, genetics, and sex. And then there's variable factors that individual may have accumulated in their career, such as comorbidities, their social determinants, and the epigenetic changes that occur in every one of us. But that's what the patient brings into the event, and that's going to impact their trajectory quite a bit. Of course, then it gets a little more complicated. We put this in the middle, and then we need to talk about the injury characteristics, the types of the body regions that are injured and the magnitude of that injury. And then there's a whole number of factors where magnitude and duration are both important, such as how much hypoxia, hyperperfusion, hypoventilation, and microbial contamination. That's going to drive outcomes in the biologic response. And then the idea that there are what we do with the patient, the facility characteristics, transport times and mode, resuscitation strategies, which is evolving quickly, and then these interventions that we do. These are going to drive the biological process and belong in that category of a hallmark. And then the early biologic responses, which I think we can divide into three when you think about it, and maybe more. But the metabolic response, the immunoinflammatory response, and the response of the endothelium and the coagulation and complement systems are the early biological responses that are so critical and worthy of hallmark designation. Now, this isn't really a hallmark, but these things all drive the outcome-based endotypes. And some of the most important endotyping work is being done here at Colorado. Chris Erickson will be talking about a little later this morning. Really phenomenal work from Mitch and his group using the omics strategies here at Colorado. Then we have the outcome side of this, which is also underappreciated and not adequately addressed. Short-term outcomes, organ dysfunction, death, the thromboembolic dysregulation that's so part of this, and then infection and sepsis. Short-term, I mean the first few weeks. And then the most important layer, which is not discussed very often, which are the long-term outcomes. Where's the patient 6 months later? If you do something early and they're still in bad shape at 6 months, that's not necessarily success. So we have disabilities that we could grade across a number of categories: impaired healing and infection. Some will return to pre-injury state, but in truth, for severely injured patients, probably none really get back to the pre-injury state. And then there's delayed death. So what we do with multi-omics is we try to capture as much of this as we can into data layers and then make some sense out of it. And so this is early days. And so what I'm going to show you today is largely early day kind of analysis in this rapidly evolving field of multi-omics. I should also suggest that any disease process can be, and for some it can be parsed out in this kind of mindset to help to remember to put into context where what you're studying relates to the big picture. So we study the immunoinflammatory response. The immunoinflammatory response is just this sliver up here of a much bigger puzzle, and it interrelates to everything, both vertically and horizontally. So there's a great opportunity in trauma. Trauma has this unique characteristic of we know when it starts. So we can define pre-injury and post-injury timeframes and periods, even epochs, if you will, where what we do is likely to be very different. There's phases pre-injury, there's phases post-injury, that early escalation phase, persistent critical illness, and then these outcomes that we just talked about. Each is ripe for intervention. You could imagine doing something pre-injury in a trauma-prone population. Simple exercise dramatically changes the outcome to injury. And of course, in the military, you need to stay in shape. Early intervention with a pathway inhibitor or even some sort of modifier, but we have to know what to do. We have to know what the biology is, and that's where we've centered a lot of our attention. And there's a tremendous promise now of multi-omics. And what we mean by multi-omics is the accumulation of high-dimensional data layers that, when integrated, give you unique insights into either biomarkers or outcomes, and requires a tremendous amount of computation. And it is the perfect problem to be solved by artificial intelligence. And we're just at the beginning of incorporating, well beyond machine learning, but AI agents and algorithms in interpretation and even displaying the data. And so embracing AI is perfect for multi-omics. And I won't claim to be an expert. I will show you an example of what we're doing here. What are the outputs of multi-omics? Well, landscape. Can you get the big picture by integrating a lot of information? We think you can, and I'm going to do a little show and tell, as I call it. Novel biology, things you wouldn't see otherwise. Target identification, model development. Your models should be faithfully reproducing the omics that you see in humans. Treatment responses, and that's where we really got our start through the PAMPER trial. And then biomarkers, to the extent that biomarkers are easy to incorporate into clinical practice. In fact, they're not. This is just I actually have to credit Angelo Alessandro for our getting into the field. His early work in multi-omics opened our eyes about a decade ago to the potential of multi-omics. He doesn't know this, but it was his papers that really motivated us to move into the multi-omics field. And this is just a summary of the body of work that we put together. More important are the areas, for example, landscape and outcomes, endotyping, biomarkers, causal modeling. We've done a fair amount of transfusion omics, and then therapeutic targets. I can't go deep on any one of these, and I don't think you'd want me to because it's Monday morning at 7:15, but I'm going to show examples of what we've been able to pull out of and some of the elements of actually getting started in a multi-omic approach to the disease process that you manage. You got to have biobanks and databases. This is a summary of ours, eight different biobanks, and some of the omics across the top and the numbers. You don't need to focus on this exactly, but it shows that you have to be an opportunist, I think. And we built some of our omics analysis on the back of clinical trials that happen to have compendium biobanks. We've done our own biobanking. I spend a lot of my time now not designing experiments, but arranging for the omics to be measured, assuring that we have the biobanks that we need, addressing biobank storage, which is extremely important, lost samples, poorly annotated samples. Even professional biobanks don't do much better in our view. So becoming a good biobanker is really important to success. And then knowing what the platforms are that you can access. And then you're thinking about data harmonization. Can you really pull two different databases together and analyze them? It's a whole different set of problems, but the opportunities are great. And we benefited from the fact that Pitt is the home of lights, which is this consortium over 50 institutions of DoD-supported pre-hospital clinical trials and early hospital trials. You can see what LIGHTS stand for. It's led at Pittsburgh by Jason Sperry, a acute care surgeon, Frank Guyette, who's an emergency medicine investigator in the pre-hospital space, and our database manager, Steve Wisniewski, in the Graduate School of Public Health. But it involves a big network. Denver is well represented in this network. And they literally get handed task orders. They don't say, I want to test this and this in patients. The DoD tells them what they're going to test. And there's a committee that decides. And they've done some of the more pivotal trials in the pre-hospital space. But it's an example of how you begin to have impact in something that is a delivery challenge. And then I mentioned to you that we built this body of work, but it's really been based on adding layers of data. So the bottom, you can see clinical data, then there were quantitative analysis of cytokines, and I'll show you an example of that. Then we got into the high dimensional data sets, and we decided to go with the commercial vendors because this is their lifeblood. They've optimized their omics. Now, it doesn't necessarily give you everything you want, but you get a lot of information from validated platforms, and there is no one right way to do this. Often you have to measure proteomics, for example, by multiple platforms in order to get the full range of information that particular data layer yields. So I'm going to start off with a little bit of multi-omics show and tell. You're going to see some heat maps and some other ways of displaying data. I'll walk you through it. You don't want to necessarily focus on specific molecules or pathways. When you get into omics, you go away from the world of specific pathways and molecules to look at patterns. And you can dive into the depths of the pathways and choose one or two to pursue. And I'll show you an example of that. But it should almost be like, a vote from the people, what you're going to do your deep dive on and how many deep dives you're going to do, because it can become quite a distraction if you spend a lot of time going down a pathway that has minimal reproducibility in humans, for example. So, PAMPER, pre-hospital plasma, improves outcomes in polytrauma patients. That was the New England study from 2018, led by Jason Sperry, and is changing practice. A biobank was created on the back of that clinical trial. We had access to that biobank, and we built this multi-layer database and began to do our analysis as we got our feet wet in a very new process. You can see 191 out of 500 patients were studied. We studied 3 time points. 0 hours is admission, 24 hours, 72 hours. That's as late as it went. First, I'm going to show you a data layer in the quantitative space. We've done multiplexing for cytokines and chemokines forever. So 21 different cytokines and chemokines were measured, and we've often displayed the data in different ways. Simply displaying it based on outcomes and as a heat map gave us the most comprehensive picture we'd ever seen of what cytokines and chemokines are doing in patients relative to their outcomes. Here we've divided the patients into 3 groups. It's not the clinical data in PAMPER is not very granular. Lived, died, how many days in the ICU did they have, and a few complications. But we divide them into early non-survivors. And this was severely injured patients transported on helicopters with plasma either on the helicopter or not. And one group got standard of care. So we've got these early non-survivors. Non-resolving, which are the next sickest, are people who stayed in the ICU for 7 days or more. The average was about 14 days. And then this resolving group, which got out of the ICU in less than 7 days. And then we've got the time points divided into the 3 time points. And I draw your attention to this time 0. And over here, we actually could cluster the cytokines into 3 different clusters. There's a pro-inflammatory set, which is up in a relative way because these are normalized Z-scores. When it's red, it's higher. When it's blue, it's lower. That's for all the heat maps I'll show you. But these non-resolving and early non-survivors had higher relative to the others pro-inflammatory cytokines, these cytokines that we had studied forever. But these patients who are still pretty sick because they're in the ICU for an average of 4 or 5 days didn't so much have an elevation there, but a relative elevation in these protective and adaptive. We call this adaptive now. You can see over time there's an evolution, but these two outcome groups separate in terms of the types of cytokines and their patterns over time. And so this is heat map sort of language. And I'm hopefully not going to intoxicate you with too many heat maps. Let's now go to these high dimensional data sets, literally thousands of variables measured by either metabolon or somologic. And I'll give you a sense. So this, all of a sudden you're drinking from a fire hose. Almost so much data you can't actually fathom what it means to have 7,000 proteins per sample across, in this case, hundreds of samples. But I'll walk you through this just to give you an idea of what it means to look at patterns. Now, we're looking at the patients divided the same way, 0, 24, and 72 hours with these different outcome groups. For 1,000 lipids, our lipids are higher at baseline, and they drop precipitously after injury. That was actually reported by David Herndon, burn injury in the late 80s, recapitulated here now in polytrauma patients, and the drop is much more dramatic in the patients that are sick. And then there's an evolution that occurs afterwards, and I'll show you just one example of what we've done here in the lipidomics side of it. For metabolites, there are metabolites that go up in the plasma with time, and then there are things such as the xenobiotics that go down. Xenobiotics means molecules produced by not the body, but another source, medications, foods, etc. And then the proteomics was perhaps the most interesting. Again, here's a we think low ISS patients are the best control. That is, patients that come in, we see them all the time, right? Helicopter or ambulance brings them in, they were in a bad accident. They have no injuries, they have a few scratches. Those are really good controls. So we've gone to using low ISS controls. But you can see massive, now the absolute values may not be so much, but the fold change is quite a bit. And these patients that do poorly have very high proteomics. There's then a resolution of the proteomics, and they evolve into different patterns over time. And the Cohen group has done some beautiful work on defining numerous endotypes based on the longitudinal proteomic patterns, and we'll hear about that a little later today, I think. Well, here's just a little bit deeper dive into the lipids. 14 different classes of lipids, time again, outcome. You can see there's drops across many of the patterns, but then there's an evolution where some of these lipid families start to emerge and are higher in patients that do poorly. One of these were the phosphatidylethynolamines, a subset of these, and you can see that here in this group of PEs, as we call them. This is a glycerophospholipid that's enriched in the inner membrane of the plasma membrane. Why it would be up and why it's there, we don't know, but I would consider that a novel discovery. We created a lipid programming score out of that, and then wanted to know whether this could separate patients into resolving and non-resolving. These are the AUC curves compared to ISS and IL-6, adding a lipid reprogramming score of multiple PIs, or even just a single, the one that was most associated with outcomes, markedly increases the AUCs, you can see in some cases, to above 0.9 for predicting who's going to stay in the ICU. Just one example, we use both internal and external validation data sets to make this a conclusion. Here's an interesting observation with metabolites. These are the TCA cycle metabolites shown here along the rows, with the columns again representing outcomes. And from the baseline state, you can see there's a broad and extensive release of TCA cycle intermediates. Now we know from the important work that's been done here with succinate, and of course lactate's been around forever, these are pretty good biomarkers. Actually, if you look across the TCA cycle intermediates are all going out of the cells, and how they get to the outside is a good question. And some of those turn out to be very good biomarkers. Now, we measure a subset of cytokines and chemokines, but there are whole families of these mediators. For example, 33 known interleukins, 47 known chemokines. We went into our proteomic data, and although this is only relative levels, we're able to find actually most of these in the proteomics. So all 33 cytokines were there, 32 of the chemokines, all the 21 known interferons are in the proteomics, and many of the TNF super family members. So we just looked at the pattern of all of these in patients. And this is what the heat maps look like for each family. And it's remarkable. In the patients that are non-resolving, they're elevated, essentially all of them are elevated on admission. So, we choose to measure a subset, but they're all there. Some will be up very high, some will just be a modest increase. But the most interesting was this increase in chemokines and the delayed increase in patients that stayed in the ICU. Very little actual chemokine research in trauma. And again, what are you going to go after? But a pattern that had not been recognized before. And then an integrated analysis. By integrated analysis, we mean taking the data layers and putting them together and seeing what you get. So we took the top 200 features from all 6 layers and put them into this correlation heat map at time 0. And you can see this big red square up here are the factors that were going up and related to ISS, correlating with ISS. This includes, not surprisingly, a lot of DAMPs, metabolites, proteins, immune mediators. We call this the systemic storm in the first paper that we put out on this. But there's another big square down here. This inversely correlates with INR. So as the INR goes up, these are things that are going down in the circulation. And we found constitutive proteins, coagulation complement. Interestingly, lipoproteins drop. So not only lipids, but the proteins that carry them are dropping. And energy lipids were going down dramatically. And this isn't a lot of computation, but it does, again, give you this overarching view of what's happening in seriously injured patients and changed our way of thinking about this. All right, so got tons of data. What are you going to do with it? We can't analyze all of it, and we're building more databases all the time. So we decided we're going to make a portal and put the data in a searchable way on the internet. It's not there yet, but how do you do that? You have to incorporate AI. Well, you go out and you find a bunch of really smart high school students who know this stuff way better than we do. And unfortunately, one of them had an uncle that was a programmer. You can see Sambo, Jay, and Bhavya right there. Sambo is the senior citizen of the group. He's now a freshman in college. And these guys are remarkable. And I'm going to play a video if it works. And it's going to be Jay, the 10th grader, describing this portal. All right. There we go. Now picking up. You know it works. We've got the YouTube here ready to go. Here's my assistant. I'll probably have to hit the escape and then find it. Anyway, I'm very, very proud of this because, crowdsourcing this information is the way to do it. And go to YouTube to find it. Yeah.
Welcome to the Pitt Surgery Trauma Research Portal, a secure, intelligent platform designed to transform high-dimensional complex trauma data into meaningful insights. This portal integrates clinical, injury, and molecular data sets to help researchers explore the biology of trauma recovery and outcomes. Access starts with a secure login. Users log in with a .edu email ID. New users request registration, which is approved by an administrator to maintain data security and compliance. Once approved, users gain access to datasets and analytical tools. Now I'll log in with a .edu email ID. After logging in, the portal provides details on the experimental approach used and empowers researchers to filter datasets based on a rich array of clinical, injury-related, and outcome-specific characteristics, enabling tailored exploration of patient-specific responses. Filters on the left let users select cohorts like Pamper or SWAT, patient characteristics, injury type or severity, and outcome measures, such as recovery time or complications. The filtered data appears instantly in interactive charts, which I will demonstrate now. The portal automatically generates high-resolution visualization. Each plot is dynamic and interactive. Hover to see data points, adjust filters, and export figures for publication or downstream analysis. The portal is also integrated with an AI chatbot. The AI chatbot supports interpretation. You can ask questions like, which cytokines predict poor outcomes? Or what does this cluster mean? It summarizes literature, explains mechanisms, and cites sources, bridging data with evidence. Let me demonstrate. The Trauma Research Portal connects data, discovery, and dialogue, accelerating our understanding of human response to injury. We are developing our trauma agent, which will be launched in the near future. Join Pitt Surgery in shaping the future of trauma and critical care research.
So AI, I think we've all gone through gene set enrichment analysis. You get these long lists of pathways. You're not quite sure which one to go after. AI is going to be the solution for that. And customized AI, so a trauma AI agent, as Jay was talking about, that is trained and then accesses very enriched sources of information that can help you with your interpretation. We see that as really the key to analyzing multi-omics. It doesn't mean that you don't do the other analysis, but this is going to be transformative. So let's move into some more specific aspects of the multi-omics. First, the analysis of the database to provide insights into subgroup-specific effects of early plasma. And for this, we did some very simple endotyping and then causal mediation analysis based on the data. Now, if you're a trauma person, you know about this trial, and that is this pre-hospital where providing thawed plasma, not FFP, thawed plasma in helicopters improved survival in a cohort of 500 trauma patients by 9%. That was considered a major breakthrough, a 9% improvement in survival. And those that survived were very sick, as you can imagine. If you take a patient that otherwise would have died, help them survive, you know, they're going to be sick. We took the Pamper multi-omic data set, just a part of it, the cytokines and the metabolite released, and established two endotypes based on these patterns. On the top, you can see there's a particular pattern, that's endotype 1. On the bottom, endotype 2. And they have different survival rates. Endotype 1 in the red on the right, you can see, just survives better, huge p-value. And now we incorporated that into the outcome and divided the patients by endotype, and that's shown across the top. Onto a small arrow now. And you can see NBI for no brain injury, TBI over here, with or without plasma. There's no difference in the outcome of these patients, whether they get the plasma or not, measured by survival. Go down to endotype 2, they survive at a lower rate. The patients without traumatic brain injury, no benefit. It's pretty clear. And but look what happens when you have patients that are in a systemic storm and have TBI. The improvement in survival goes from 9% to almost 70%. Now the numbers are a little small, but you can show this with the bigger numbers. So quite dramatic. So omics helped us understand who benefited from this therapy. And I suggest that this is more important for negative trials. If you understand why and I think you'll probably find subsets that probably do benefit from your intervention, but you'll understand why it fell because perhaps you didn't change what you thought you were changing. We also could see that giving plasma to these patients with different injury patterns resulted in the metabolism of the plasma in a different way. These are two heat maps. What's on the actual molecules don't matter. It's just these patterns where these arrows demonstrate the patients that had TBI and got plasma compared to patients that didn't have TBI. And you can see they have very different proteomics and very different lipidomics. So they just use the plasma differently. And that's an important kind of principle that came from this work. Now we asked, could we use mediation analysis to identify what it was in the plasma that resulted in an improved outcome? Big table, you don't have to focus on hardly anything, but it's the patients divided into those different groups with all their characteristics. And of course, there's difference in their characteristics. But the key thing is this survival, where the dramatic improvement in survival was identified in the E2 TBI patients. So we wanted to know whether we could find the molecules that might explain the difference. And we worked with one of our computational scientists to use a combination of linear regression, and which is a univariate approach, and a multivariate approach, looking for both direct variables as well as latent variables, which they all tell us you need to know. These are the ones that are inferred by the variables that you do have, and ran these through mediation analysis, a common technique to try to understand the causality of the intervention, which in this case is either administering plasma or not. I'm going to not take you through any more of the analysis other than just to show you what we found. We found molecules that were associated with clotting and fibrinolysis, and also lipid transport, lipid mediators, and possibly some that are related to brain protection. We're particularly interested in this serpin A5, which is a protease inhibitor, the first of the serpin family to be identified, originally described as protein C inhibitor. Many of these block protein C, but this was the first to be identified, and kind of makes you think, are we replenishing a drop in this particular serpin with our plasma? We know that APC is activated. It has a lot of positive things, but one thing it does is it halts blood clotting. And it's a mechanism to shut blood clotting down after it gets activated on the endothelial surface, so you don't overdo it. Or imagine that happening in somebody's brain after they've been injured, where another 10 or 20 cc's of bleeding might be the difference between life or death. So we hypothesize at least that the serpin A5 may be blocking the overactivation of APC in the brain and preventing that bleeding and progression. Hypothesis generating, no proof that that's happening, but we got this out of causal mediation analysis. Of course, we'd like to have a biomarker to know which patients fall into that E2 TBI group. So we took a group of 30 variables that were measured in a quantitative way and looked at which are most predictive. And we found, not surprisingly, some brain injury markers like UCHL1, and then endotheliopathy markers, inflammation markers, put that into an AUC analysis. And you can see we can get pretty high when we combine UCHL1 in the blue line with syndecan1 and a couple of inflammatory markers, AUCs of well over 0.9, in predicting who will be an E2 TBI patient. And this is being taken into the clinic now. This was already going before we did this analysis, but they're looking specifically using a point of care analysis of UCHL1 and GFAP to identify patients that might benefit from plasma. Now, another thing we had an opportunity to do was to directly compare plasma administration with whole blood. Now, I naively thought, if you give whole blood, you're giving plasma, so should you have to give plasma as well? And interestingly, through an analysis done by Lights, which was designed to look at the consequences of different transfusion approaches, to do some omics. And even though it was a very complicated analysis, we were able to identify patients that got component and no whole blood, which means they got plasma, and people who got whole blood and didn't get plasma. Took a lot of additional computation. We did omics on 134 out of 1,000 patients that were enrolled. The reason they got patients that got transfused is all these patients had to go from the ED to the operating room, which means they have a very high likelihood of getting transfused. And these are the steps. Hamid Mohammadi took this on. I'm not going to go through the steps, but just give you the output. And that is that the proteomics demonstrated that patients that got plasma but not whole blood had an upregulation of a module of protein shown here, all related to coagulation and fibrinogen, and you can see the clotting factors, and then a suppression of certain platelet factors that was not seen in the patients that got whole blood. Here's the plasma for the what we call the clotting factor module. These are, we measured donor plasma, that's here. Here are the whole blood patients. Here are the component or the plasma receiving patients. And there was an effect that was particularly pronounced in the trauma brain injury group. The platelet cohort of proteins suppressed in the plasma patients down to baseline or even lower than baseline levels, not seen in the whole blood patients. Here's the whole blood patients. Neither one of these changes took place. So this says that plasma in whole blood is not the same as getting plasma when separated into components. Chris Baird and others are doing work showing that the cells probably release factors that change the plasma constituency within whole blood. And it suggests, does not prove, that giving plasma in addition to whole blood with patients with TBI may be something we'll do in the future, or maybe we'll understand what it is in the plasma and we can give the specific factors directly. Okay, so here's the, I think, last or near last omics vignette that I'm going to share with you. So I showed you that there's this massive release of cellular content. So we wanted to know, what was causing that? Could it be targeted? Could we change the trajectory potentially, at least in experimental systems, if we suppressed that massive release? And to do that, we needed to reverse translate our observations into an experimental model, something else that I think we need to use omics for, which is to validate our models. So reminding you again that in the metabolite side of this, that there is a big release of metabolites and a big release of proteins. So the kitchen sink, so to speak, ends up in the circulation. So we hypothesized that one of these versions of programmed cell death, and if you're in the field of almost anything anymore, you know something about programmed cell death. You've heard of apoptosis, but then there's lytic forms of cell death, necroptosis, pyroptosis, ferroptosis, each a separate pathway, each with separate effector pathways. And we designed an experiment to ask this question. And I'll draw your attention to this protein here called gasdermin D. It, when cleaved by inflammasomes, forms a pore where molecules can get out of the cell, and eventually the cell lyses. And this is how IL-1, for example, gets out of a cell. And so I'm kind of giving away the punchline, but it's better to do that up front so the rest of it makes sense. And we've been working with mouse models forever. We'd shock them for 90 minutes, we'd shock them for 2 hours, we'd add tissue components to it. It's really hard to make a mouse sick. They either get modestly sick or they just die. But we decided we would determine what it took to get the similar release of TCA cycle intermediates out of a mouse into the circulation. And we use, you can see aconitate, succinate, and malate here. And this shows that in a dedicated assay for this, an HPLC assay, to get the same TCA cycle intermediate release, we had to shock the mice for 3 hours. Not surprising, I guess. These are animals that live in very harsh environments. These young mice tolerate shock very well. In order to see the TCA cycle intermediate release and design a experiment shown in the next slide here, using either inhibitors or genetic approaches to block these pathways. These are heat maps of a full metabolic analysis in mice done at that 3 hour time point. And I'll walk you through, hopefully you can see my arrow. Here are the control metabolomics, and you can see them divided into different classes if you want to go into that depth. And then here are the shock mice, the vehicle controls that are needed to administer agents are shown there. And we see a very similar release to what we see in humans. And metabolites, there's a suppression in some lipid levels. And on this side, we have the inhibitors. On this side, we have a couple of knockouts related to these pathways. We have an inhibitor for apoptosis, necroptosis, ferroptosis, and a pyroptosis inhibitor known as disulfiram that I'll talk about a little bit more. Here, you can see that all of the cell death inhibitors reduce the intensity of the red bands, which means there's some inhibition, but the most dramatic was with disulfiram, which was not subtle. Sorry for all the jumping around here. So disulfiram caused about an 80% reduction in the release of cellular contents, and also reduces some of that consumptive pattern that I showed you as well, but I'm not going to talk about. So we took it over to the knockouts. Gasdermin D is that effector molecule that leads to lytic cell death with pyroptosis. The Gasdermin D knockouts look just like the disulfiram. MLKL, which is a effector for necroptosis, didn't have an effect. So at least in mice, it looks like that early release in the setting of severe shock is at least partially driven through programmed cell death or regulated cell death, known as pyroptosis. Well, what is disulfiram? Well, if you're around long enough, you know that it's antabuse. It's the drug that we give people with alcohol use disorder and makes them violently ill, so most of them won't take it. So it's not that effective. But it was shown in a nice paper 5 years ago to block that pore formation. Now, it does dozens of other things. So the obvious question is, can you give disulfiram and improve survival? And here in an experiment where the disulfiram was given either pre-shock, just after shock started, or 3 hours later, in each circumstance, it significantly improves the 7-day survival of mice. And so, at the talk for the research retreat later, I'll go into this in much more depth. But this is the graphical abstract that went along with the STM paper that came out earlier this year, showing that disulfiram could be used, although we don't think it would be a good choice because of its off-target effects, but could be used off the shelf now in a possible clinical trial. The other thing we found is that this pathway is far more active in females. And when we did our mouse experiments, we did equal numbers of males and females just because it's the right thing to do. And we found dramatic differences between males and females across a number of our parameters. No big surprise, but the field, research for years has kind of ignored that. So this is the end of the talk. Hopefully I've convinced you there's a lot to learn from multi-omics. You need to build biobanks and develop the computational efforts. You have that here. You've got people who really know how to do this, and trauma is wide open. There's a lot of data layers left to be added to this kind of analysis. It's a big group at Pittsburgh. Many of these are computational scientists. Many of them are surgery residents. We can talk a lot about how to train a generation of surgeon scientists comfortable with this kind of biology and study. A key partner in all this has been Todd McKinley, an orthopedic trauma surgeon who is very good, interested and good at the basic science side of this. And he's been a great partner through his consortia and metric, and many of our conversations emanate from the work that we're doing together. So thank you. It's a pleasure to be here. Hopefully I didn't go too deep on any of this. I warn you that next talk goes a little deeper, but still keeps it hopefully relevant. Thank you.
Billiar, thank you so much for giving this talk and summarizing, decades of work and I'm also pleased to see Jason Sperry, a big part of this. He was a resident when I was a junior faculty member at Hopkins.
Yeah.
I'm sure he's a great team member. So I have a couple of questions. I don't know if we'll get through them all. Maybe I'll ask them throughout the day. But in terms of these markers that you're picking up, which ones are causing the ongoing injury versus which ones are just a byproduct of the injury? And I'm sure you ask that question every day because you're looking for points where you can intervene, like the disulfiram. But I mean, isn't that really the next step to figure out where in the cause and effect spectrum that all these markers are?
So it's a really good question. Take a little bit of an agnostic approach to this. Biomarkers don't have to be related to the causality. If they are, then they may be a better biomarker because then you can be confident that your intervention might be more effective. IL-6 is an example of something that we all think would be an interesting biomarker and probably target. But some of these we think are just byproducts of the biology that's taking place, such as the cell death. We could probably measure the cleaved form of gasdermin D, for example, in the circulation and be even more confident that we might want to intervene on a patient with a hopefully someday selective gasdermin D inhibitor. But we're a little agnostic right now about the cause and effect. The phosphatidylethanolamines, for example, no clue why they're being released. We're working with lipidomic experts to kind of help us think through it. But right now, it would just be a biomarker. I think we can talk about biomarkers for a long time. It's a lot better if there's an existing assay for it and you don't have to develop. So there's the ease of use biomarker argument. The idea that the biomarker is part of the biology and therefore would be even better because it's going to identify the patients that have the biology. And then how many biomarkers can you really measure and how quickly can you get the data back? Because most of what I'm talking about are things that you're going to want to do within the first, 2 to 3 hours. And, for some of these things, dedicated assays already exist. Others will be very difficult to develop assays.
That was actually going to be my next question, comment. And this is probably more for Angela D'Alessandro, right? I mean, ideally what you want is a dipstick, that you can take somebody's blood and dip the dipstick and get the result back in 5 minutes. And I'm sure you and your collaborators are working on ways of tightening the turnaround time, right? Because, these biomarkers don't do you any good if you get them a week after the injury, but if you can get them a couple of minutes after, then I think they'd be much more.
Yeah, that's a whole great opportunity. And then how do you test them and how do you convince the world that these are going to be effective? And if we go back to that early slide where I talked about the hallmarks and the outcomes, for example, what outcomes are you going to map it to? Where live dead is good, but even if you do an early intervention and the patient's still completely dysfunctional a year later in a nursing home or skilled facility, did you pick the right outcome to map the biomarker against? So I think there's a lot of opportunity.
That was an amazing talk and definitely something that Japan are looking into. You had alluded to epigenetics on a couple of your slides. And so I was curious if you often work looking at epigenetic clocks and how that interacts with the premature physiologic aging.
Yeah, great question. How old is the patient actually, from biological point of view? We haven't gone into that. I think that's a great opportunity. It's one of those pre-injury acquired factors that would perhaps help us and should be measurable. I will present just a little bit of information later on our ATAC-seq, which is the only epigenetic related work we've done on circulating leukocytes. So, I think the pre-injury state as a data layer, a lot of opportunity, and certainly their epigenetic age is likely to be important.
Thanks for an outstanding talk. Thanks for being here. It's really wonderful to collaborate on a lot of this work. What about comparative biology across other threats and disease states? You know, the funding agencies are talking about threat X, whether it be trauma or burns or sepsis or the next pandemic or even cancer. And there's probably common biology and probably a separate biology. I'm wondering what your thoughts are in that regard. Have you compared other disease states and how adaptable are these techniques?
Yeah. So I'll broaden the question and then I'll narrow. Broadening it is there is a sea of databases out there. And sepsis always gets studied faster and to a greater extent than trauma does. It's just because there are more people studying sepsis. It's not just a surgery, EM sort of specialty. And then COVID got studied like crazy. I mean, there are so many COVID databases. So it's easy to take your signatures into these other diseases and determine whether there's overlap and what the differences might be. Trauma just has a certain dearth of high-dimensional data sets, in large part, again, because I think it's a smaller community studying trauma. But yes, the PE patterns are there in COVID. The PE patterns are there in sepsis. Interestingly, mannose, the sugar, it's almost identical to glucose, is up and is up much higher in people who do badly. And there's no clue on why that's up. But you can see it in sepsis, you can see it in trauma. It's one of those molecules you'd have to decide you want to dedicate 2 or 3 years of your lab's efforts towards understanding why mannose is up. But you do need to work with the experts. So you have to find, a glycobiology expert that's willing to think about it with you and possibly work if you wanted to go that direction. But it also says that within these data sets are hundreds of NIH grants. If you could just develop the model systems and the translation back and forth from human and sepsis, COVID, burns, all represent the lateralization, I call it, of the observations.
Any other questions? That was fantastic. Thank you very much. We'd like to give you some university bling to take with you or get sent to you, I suppose. And just want to let the audience thank Dr. Billiar for this talk. Stick around. The residents are going to meet with him for a while. Then he'll meet with some other folks. And he will be at our research retreat if you have any other questions, beginning at 10 o'clock over in AHSB. Thank you, Tim.
And all complete the wellness survey. Our departmental percent completion is less than 20%. So we have to boost that up. We'll send around